So I'm very happy to be here. I work at Philip Morris as a scientist there, and it's my first time in Open Talks. And as I said, I'm very happy to be here to see people with many different backgrounds, but a few common objectives. That is trying to improve the ways that we do predictive toxicology. So what I would like to do is give you a bit of information about some of the challenges and some of the lessons that we learned about sharing, data sharing. And this is something that is very important to us because we want to be open with the community, we want to share our data, we want people to review our data, but we need to define exactly how we do this, at least in the case of high-throughput data or omics data. So what I would like to do is use my talk to set up a bit the stage, give a little bit of background, give some introduction to the speakers that we will have throughout the session, and as I said, share some of the things that we're doing at Philip Morris, and hopefully to see as a starting point for discussion. Some of the challenges we face, maybe they are the same, maybe you have others, and we can comment on that. So just to start, there's an old saying that says that the picture is worth 1,000 words, and usually this refers to the notion that complex ideas can be better explained with an image. And if you bring this into biology, which is a pretty complex subject, it turns out that we've been doing this for centuries. And this is one of an early example. This is a picture by a Spanish neuroscientist and Nobel laureate, Santiago Ramón y Cajal, who was made around the 1890s. And this is what Cajal used to do. He used to use microscopy, try to describe biology in the way of a collection of drawings that's part of his legacy. And if you look at this is for an example of retinal neurons. You see there's a lot of information there. And it's a very good representation of reality. And actually it is quite impressive, especially if you take into account what passed by state-of-the-art microscopy by the 19th century. But this type of representation has some limitations. On one hand, the information that it has, as much as it is, it's only qualitative. We know what's in there, but we don't know how much is in there. And also the other problem is that this is incredibly low throughput. One can only imagine how long it would take Cajal to generate one of these pictures. Fortunately for us, throughout the 20th century, there was a series of developments that allowed us to increase or to improve in the way we do microscopy. With the development of fluorescent labeled antibodies and then confocal microscopy, we were able to reach a higher level of resolution and also to do some quantitative analysis, so we know not only what's in there, but how much. But still, these techniques, they still have the problem of low throughput. It takes quite a bit of time to process and analyze each one of these images. Then finally, by the end of the 20th century, they started developing high-content screening systems. And this allowed us to do a quantitative analysis, but in a much rapid way, a much higher throughput. Which means that we passed from this idea of a picture worth 1,000 words to have the ability to process thousands of pictures in a short period of time. And the amount of information that you can get there, it's actually priceless. Now, if we put all the tools that we have here in perspective, we will have on one side of the spectrum tools that give a lot of resolution but low throughput, like microscopy. Then you have things like high-content screening, also known as high-content analysis, which gives you much throughput but keeping a good deal of resolution. And then we have other tools that are commonly known as high-throughput screening, where you may not have as much resolution, but you have quite a bit of throughput. Now, between high-content and high-throughput, there's quite a bit of overlap, there's some differences, but some similarities. And for the purpose of this talk, I would like to group them together, because I think in terms of how we share data and how we process the data, both type of techniques will share similar types of challenges. So I would like to go one step higher and talk in general about, say, high-throughput, high-content screening, defined as any technique that can automatically detect and quantify a large number of biological readouts in a short period of time. Keeping in mind that this has only been possible because there were advances in molecular biology, but also robotics, microfluidics, and also bioinformatics and computational tools. And these techniques are very versatile. They have been used, they are currently being used for many different things. They started out in pharma for drug discovery, but also we do things like functional genetics, and even in academia, things like disease research. And a little later, it started to being acknowledged by the field of toxicology. And this was, it was somehow made possible because regulatory bodies both in the US and also in Europe, they started issuing a series of documents that they encourage the use of novel technologies to change the way we do toxicity assessment. Technologies that would include high-throughput scheming, but also systems biology, omics technology, and other computational tools. And the idea, as you are well aware of this, is to move away from that traditional type of assessment, using large numbers of animals exposed to really high doses and then try to predict what will happen in human populations, to a more mechanistic based approach in which you use in vitro data to try to predict human outcomes by the development of these AOPs. And there's some initiatives on that sense. It would be a nice way to prioritize chemicals and select those chemicals that need further study. And also, this view copes with one of the issues, that is the large use in animals that are required for traditional toxicity testing, which raises a lot of ethical issues. As I said, there are a few initiatives at both sides of the Atlantic. We have the ToxCast and the Tox21 programs in the US, the Suarez and the EU ToxRisk. I just removed the 21 because I was told this morning that it's too much, so well done. And I would like to use this slide also to serve as an introduction for some of the speakers that we'll have today. So we have people from the APA, and he is going to tell us about the ToxCast, how they do the data processing, and hopefully some of the challenges and solutions they will find. And from Europe, we will have Barry. He's been leading the ToxBank, and he's going to be talking about how he used ToxBank for the Suarez compounds. Now, before we listen to the speakers, I would like to take a few minutes to share with you some of the things that we're doing with Philip Morris. Philip Morris is a tobacco company. We're interested in new tobacco products, and we do quite a bit of toxicity assessment, actually. And some of the things that we've done is to develop a workflow for systems biology-based toxicity testing, which is very much in line with this idea of the toxicity in the 21st century. So I would like to share with you what we do and some of the lessons that we learn and some of the challenges. So this workflow, it's a 5-step process. The first step, as in any experiment, starts with data generation. So basically you select what is that you want to test, you select where you want to test it, and we use mostly 2D cultures as well as 3D tissues, and then you decide how you want to test it to address your scientific question. What's the exposure, what's the doses, the time points, etc., etc. And the idea is to try to generate as much data as possible and come up with this system response profile. So basically what we do is we use high content screening methods, and then we use also omics, mostly gene expression, but also proteomics and metabolomics, to try to come up with some information on some mechanisms of toxicity. Now at this stage, and if you focus, for example, in gene expression, what you will get, if you're lucky, is a long list of genes that are differentially expressed between your exposed and non-exposed cells. But there's not much mechanistic information out there, and actually it's really hard to try to discriminate what is the true information, the relevant information for all the experimental noise. So in order to try to shed a light here, we use an approach that is based on network biology. So basically what we have done is throughout the years develop a series of biological networks that try to represent key processes in the cell, proliferation, inflammation, etc. These networks are being built based on the review of the current knowledge in the literature. They have been published, but they also have been shared with the community, with the scientific community, so they can be reviewed, they can be criticized, and they can be improved to try to come to an agreement as to what are the best possible networks that we can build. And the idea is that when you input your gene expression data into these networks, you can start highlighting those molecular pathways that are perturbed upon exposure and start getting mechanistic information. Now, the beauty of these networks is that they are computable. So not only you, excuse me, not only you can identify which networks are perturbed, you can quantify the level of perturbation in those networks. And you extract a lot of information, and when you put together information of all the biological networks together, what you end up is with a biological impact, with a factor, with a measurement of how much is the impact of compound X in your system. And where do we use high content scheming? We use it at 2 levels. We use it at early stages because this allows us to select those experimental conditions that are more relevant, and they make more sense to go to further analysis via transcriptomics. But we also use it to confirm some of the predictions that we get out of this network biology with gene expression data. So if I say my prediction is that compound X increases oxidative stress in my system, well then I can measure, for example, glutathione levels on the cells and see if there's a decrease that will confirm the effect. This is just to show that these networks were originally generated to test tobacco products, because this is a tobacco company, but the workflow can be used to test any other thing that you can imagine. And we have done that. We have used this to analyze environmental toxicants, HPLCs, things like nutraceutical, etc., etc. And by doing that, we have learned quite a bit of things, and we have faced some challenges. And the first thing that we notice is the issue with the data overload. Generating data, it's relatively easy. Processing and analyzing the data is a whole different story. It's large volumes of data. You have a series of challenges. On first hand, what do you do with the data? Where do you keep it? How do you archive it? And second, how you process and mine the data? How you extract the relevant information? And that brings me to the second point. That is that it's QC. With these approaches, and this is valid for gene expression as well for high content or high throughput, not only you have to ensure the quality of your data, but you have to ensure somehow the quality of the processes that you use to generate your data and to come up with the conclusions. And in our hands, what we thought is that one way to QC the data to ensure the quality of the data is very simple. Is you do multiple biological and technical repeats. If I do a measurement and I predict oxidative stress, I put one next today, I repeat the experiment tomorrow, or next week, do I get the same result? We put it together with the statistics, and we can come up with some statistical power. Now, how do you ensure the quality of your processes? Well, in this case, what we think is that crowdsourcing initiatives are the way to go. And one of the ones, one that we got involved in, it started as a collaboration with IBM, is this SBM Improver, which stands for, and I have to read this because I always forget, it's Systems Biology Verification combined with industrial methodology for process verification in research. And behind this very complex, it's just an effort to try to verify some of the tools and methods that industry uses in terms of systems biology. We have done this at the gene expression level, for example, through challenges, trying to identify or to compare different algorithms that try to extract diagnostic signatures, cancer, COPD, etc., and disease. We have done this to try to compare how translatable it is in vitro to in vivo data or mouse data compared to human data. And we have done this also to improve the biological network. So there are different options here. In the case of high content screening, we haven't done that yet, but we're currently working with Barry Thomas and their team to try to come up with some challenges or some ideas of how to verify, ensure, or improve the quality of these processes of high content screening data. But that brings me to another point, that even if you want to do that and you want to share your data, there are some challenges on how you do that. First of all, where you should put the data, what type of data you want to share, what format, should it be the raw data, the process, and as important as the data is the meta information, what type of meta information you put there. So, in order to set the stage and to brainstorm a little bit on some of the things that we can do to accomplish these things, one can think of, okay, if we have the ideal situation, so the perfect database that people can upload and download information, what type of data should we put there? And we have mainly two different options. We can put images, or we can put data, numbers. If we have the images, whether they are raw or they are processed, well, the advantage is that this is the real raw material that comes from the machine. It's raw as it gets. And you can actually validate any different stage of the process. Even if you want to validate whether your antibody is sustainable, supposed to stain, you can even do that. The problem is that these result in very large data files, and there may be some issues. There are some initiatives that try to cope with this, and there are some ways to share data, images from high content screening, but still the question would be if this is really necessary and if it's really worth it to make this. The other option, as I said, is to share the numbers, whether it's the raw data, the process, or a combination of both. Well, this has the advantage that you require less storage space, You don't really need an image analysis software to extract the numerical values, so it increases a bit the complexity. But in some cases, especially if you only work with process data, you have the problem of how this data was generated. And this is particularly concerning when you use commercial software. For example, there are black boxes. You input the data on one side, you get your process data outside, but you have no clue how the data was processed and what happened. And then, as I said, as important as the data itself, it's the metadata. This is something that we discussed yesterday, and I would like to emphasize, because I think this is very important. But what type of metadata should we include? One way to address this is, well, I think you need to answer these three questions: the what, the why, and the how. You need to answer the what. What are you testing? Which compound? Where? Which cell? Or organism? What are you doing? Which assay? Which endpoint we test? And where this comes down to in the end is that we need an ontology. We need to be able to define what we're doing. And ontologies in terms of high content screening, they are not completely well defined. And this is important, and it's not a trivial task, because at the end of the day, if I'm talking about an apple, I want to make sure that everybody out there understands where an apple is, and they don't think it's something different. The second thing that we need to show or to include is the why. What's your study objective? What was the study design? Because having a good rationale, it will allow anybody else to see where this data set was generated in the best possible way, and if it's fit for purpose. And then finally, it's important to address the how. What are the materials that we use? Where they come from? If they're single compounds, if they're complex mixtures, how they were generated. The cells, what type of cells? Primary, where are they coming from? Very important, the methods. How was the exposure taken? What are the numbers of repeats? Extremely important, a good assay description. Assays are complex, are differences, and an assay is not always the same. As I know, when you talk about a car, you want to make sure if you buy a car that you're buying a Lamborghini and not a 40 or a Volkswagen. Very good shape. The same with the assays. You want to know exactly how the things were done. Also, we need to talk about the data processing. How it was processed at the different steps, how you go from the raw data to the process value. And then we need to talk about some things that biologists sometimes forget, and I include myself in this group. That is the statistics. We need statistical power, and we need to provide enough information so that bioinformaticians or computational biologists can do the work. And things like plate layout, for example, is something that is extremely important, because we need to know whether there are batch effects that may hamper some of the conclusions that we take of the data. Now, I also want to take this slide to kind of an introduction for one of our speakers, for Yang Wang. She's working at PubChem, and she's going to give a talk on how to what's the practice that PubChem has for hosting these types of data. Hopefully, some information on what makes sense or doesn't make sense to include in your systems. And to finalize, it's a question of the why. So I share some ideas of what we can do, but then the point is why we have to do this. Is a big effort. Why we should do that. One thing that comes to mind is this change is hard, and new approaches often face skepticism. We are changing the way that we do toxicology. We're changing things that, or we're trying to change things that have been written in stone for decades. And that takes time, and that makes people, especially those that are not so familiar with the technologies, to be skeptical. And believe me when I tell you, I work for a tobacco company, so I know a thing or two about skepticism. But the idea, or what I think, is that if you want to cope with skepticism, if you want to avoid it, the best way is to be as open as possible. So you have to try to be as open as possible with scientific methods of the data, to facilitate, to encourage, to foster the scientific review of the data and the approaches and the findings, and ensure full traceability. Now, in order to do that, we need guidance. We need to know exactly how to do that. We need at least to try to agree on what's the best way to do things. Because that's the only way that we can increase the availability of our data, that we can facilitate the comparability of our different studies, and foster reproducibility and reliability. And one last point that I would like to make is that we're changing the way we want to do toxicology, and maybe we also have to change the way we think about reviewing the scientific data. And try to complement the classical peer review system with more initiatives of crowdsourcing, what is called as the power of the crowd. And I'll give you an example. If you think about the classical peer review system, basically you have a scientific question, you generate your data, analyze your data yourself, and then you write a series of conclusions of claims. And then you send that to review, whether it's a paper, a grant, etc. And the reviewers, what they pretty much can evaluate, it's limited because they don't have access in general to the full data set, only what was used to write that paper or that grant. So they can check things like innovation or the logic, whether the conclusions that you have in your paper are supported by the data. But we don't have the full information, and that may lead to issues in terms of reproducibility, etc., etc. Another thing that we can do is the crowdsourcing, something that was the basis of the improver that I just mentioned, but there are other initiatives, like challenges, etc., etc. And it's at some point in the process, we can think about sharing the full data sets with the community so that they can be independently analyzed. So that then we can all get together and discuss the results and try to come up with conclusions that are seen as valid or approved by the majority. And this sort of dialogue will help to increase the trust and to reduce the skepticism. And it will be a nice way to check the quality of the R&D methods that we use, both in academia and in the industry. And that's all for me. I just want to leave one take-home message, which is that it doesn't really matter how many resources you have. If you don't know how to use them, they will never be enough. And I think it fits very well with what we're trying to do here. We have these technologies, extremely versatile, are destined to change the way we do things, but we need to make sure that we use them wisely and we use them in the best way possible. And I think that learning how to share the data and enforcing it is the way to go. And with that, so thank you very much for your attention. I will have questions.